दिख रहा है?
Yes, yes.
So from basic we are starting. We don't everyone know what is RNA, but there are many types of RNA. We have categorized. We have not all of us have are knowing that RNA is categorized into coding and non-coding RNAs. So in coding we are knowing mRNAs are there. For non-coding there are there are many categories. One of them is very we are knowing very well that rRNA, tRNA are there, which is a very crucial part of transcription and translation. Another category of non-coding RNAs is small RNA and LNC RNAs. In the category of a small RNA, we have many types of RNAs, and we are very well aware of them. Some of them are microRNAs, miRNA, siRNA, snRNA, SNO, tRNA, HCRNA. It is there are lots of RNAs, and every day we are identifying and we are increasing the list of these small RNAs. Another class of RNAs is the long non-coding RNAs. The basic difference between these small RNAs and LNC RNA is about their size. So we are knowing that messenger RNAs basically are the which are carrying the genetic code, and only due to this messenger RNA we are able to translate our genetic code into a protein. But for these coding, there are many supporting RNAs also there which help them to get translated, and we are getting the proteins. Some of them are ribosomal RNA and transfer RNA. Without these two, we will not get the protein basically. And these ribosomal RNA are the catalytic are the part of the catalytic component of ribosomes. Means the ribosomes are comprising these RNAs and some of the proteins which are forming a nucleoprotein complex, and these nucleoprotein complex helping to synthesize polypeptide chains with the help of tRNA. And we are knowing that tRNA, which is transfer RNA, they are carrying the specific amino acids for the triplet codon. And these tRNA are going to bind on the ribosomal RNA, and they are with the help of messenger RNA, which has already bind to the ribosomal RNA, they are able to transfer the genetic code into the amino acids. Along with them, there are small nuclear RNAs. These small nuclear RNAs are basically uh, specific proteins which are associated with the RNAs and forming a small nuclear ribonucleoprotein. And they are helping in the process of precursor mRNA. Means the mature mRNA, the biogenesis of mRNA, and there are two steps: precursor and primary and precursors. Then these are helping to get a mature mRNA. Along with there is small nucleolar RNA. These small nucleolar or RNA are also responsible for sequence specific nucleotide modifications. Another class is PV interacting RNA, piRNA. We are knowing very much, and these piRNAs are basically playing a role in gametogenesis, where it stabilizes the germline cells. And along with this, is another class of small RNA is the microRNA. These microRNAs are well studied now, and lots of people are knowing about microRNAs. And every day we are getting new uh, results, new discoveries with these microRNAs, and these microRNAs are helping a lot, and they are actually participating in the post-transcriptional or post-translational repression or regulation of genes. Basically, small RNAs are means their size is very small. It may be around 24 nucleotide long. And similar to microRNA, there are tRNA, siRNAs, which are also helping in the regulating the regulating the expression of genes, yeah, proteins for their activity. Along with them, there are another class of them is the long non-coding RNAs, which is LNC RNA. We are knowing the main thing is that though these RNAs are are long up to 20 nucleotide long, but they are unable to get translated into a viable protein. Means the proteins are not stable; they are get degraded very soon. And the important thing is that we, after human genome sequencing, we are knowing that only 2% genes are going to encode a protein. What about the rest? So it nowadays a large number of LNC RNAs were discovered, and they found that the portions which are not coming under the gene, or the portions which are going transcribed but not get translated into gene and into the protein, comes under this category, long non-coding RNAs. And they are started finding that they have also role in the expression of genes. Basically, till now we are knowing that they are controlling epigenetically or post-transcriptional level, and they are controlling the regulation of genes. Still, the origin of long non-coding RNAs is a matter of debate, where very few of them are knowing that very few processes is known about the uh, origin or evolution of long non-coding RNAs. So today's my topic is microRNAs. Basically, I did my all research in plant related to microRNAs. So we will talk about more microRNA, plant microRNAs. So the first question is, why microRNAs, and how these microRNAs are evolved, and why they are here to control the gene expression. So this is actually, till we are not knowing what happened, why these mRNAs are there. We are knowing the regulation of genes were controlled by so many factors. We have 5 prime UTR, 3 prime UTR. In 5 prime UTR, many small motifs are there, and these motifs are able to help in controlling the expression of genes, either in the form of activator, enhancer, or other type of motifs which were controlled by transcription factors. And these transcription factors are very much efficient in controlling the expression of genes, many genes or co-regulated genes inside an organism. But still we found the regulation of genes are also uh, controlled by other mechanisms. One of them are the microRNAs. Basically, microRNAs are small RNAs which is of size 20 to 24 nucleotide. Either they are regulating the genes by cleaving their target mRNAs, or they are inhibiting the translation of mRNAs. And we know that these microRNA were first discovered in C. elegans, and after that, lots of microRNAs were discovered in animal system. In plant system, we have also microRNAs, but there is a little bit difference between their biogenesis. Here we can easily able to see the differences. There are genes which are going. This is the important aspect from where microRNAs are coming. Means either it is a part of a gene, or it is a part of a fragmented gene, or they have a separate gene for that. So microRNAs have their separate genes. Then these genes are able to transcribe and transcribe into the microRNA through these processes. So similar to the mRNA, it is also forming first time primary miRNA. The main thing of miRNA is that they have a stem loop structure. Basically, stem loop structure and two overhangs which are very much complementary to each other. This complementarity is able to help them maintain a structure of stem loop, and the process while removing this stem loop from the primary miRNA is the main mechanism. So if we are here, we can see that these primary mRNAs first time, which is which is also having a poly A tail, is going to be into a duplex format. Means they form a complementary duplex form with the help of proteins, which is DCL1, HYL, and serrate. These proteins are in combination are helping to remove the stem loop structure and the non complementary part of the primary miRNA. And after this, the duplex structure of this miRNA is methylated with the help of HEN1 protein. And this methylation help it to transport from the nucleus to cytoplasm, where it make a complex with the ego gene, which is known as also known as dicer. And these ego genes are helping these duplex to separate into a single part, which is complementary to a target gene. And these miRNAs are get attached with the target gene to stop its activity, or by either degrading it or by hampering the translational process. Similar to plants, animals also having the same, almost same mechanism, but there are homologs of DCL1, HYL, and serrate gene, which here we know as DROSA and PASA gene. These DROSA and PASA genes are helping in cropping the stem stem structure and the non-complementary part of the primary mRNA. And these are uh, cropped by these combination of these proteins, DROSA and PASA, and they form a precursor miRNA, which is here. There is no any homolog of N1, but the structure is intact. Stem loop structure is intact, and this is able to help in to get transported into the cytoplasm from the nucleus with the help of exportin 5 protein. And in the cytoplasm, dicer with the help of other proteins make a complex, risk complex, which is able to uh, produce a mature miRNA, and these mature miRNA is get attached with the target gene to silence them. So the basic difference between these two in these two systems is that there are homologs of DCL1, HYL1, and serrate, and in animal we have DROSA and PASA, and there is a no HEN1 protein in animal system. They have stem loop structure intact, which is get transported into the cytoplasm. But there is a major difference in the mode of action of microRNAs. The major action is the major difference is that plant microRNAs are highly complementary to the target genes. Means the entire length of the microRNA is get attached with the target gene, and this will help them to cleave the mRNA or target gene. But if we talk about animals, in animal there is a seed sequence which starts from the position 2 to 8 position of the microRNA, and these seed positions are complementary to the target gene. So when they attach with the target gene, it will not only not, it is not going to cleave that particular gene, but they are able to stop the translation of that particular target gene. In this way, they are regulating the expression of genes in animals, and the complementarity is very high in plant rather than in animals. Though, so the efficiency or the affinity of microRNAs in animal is very less than the plant microRNAs. And the importance, basically the importance of microRNAs are that they are very much involved in the plant growth and regulation and their development. So a lots of work have already been reported or done by the scientists, where they find so many microRNAs which are playing a very crucial role in the plant growth and regulation. Some of them are listed here, and these are the very known microRNAs. Here I have listed the microRNA 156, which is going to target SPL family genes. These SPL family genes are playing role in leaf development, root development, secondary metabolites, in ZMAs it is, and Arabidopsis also. Similarly, microRNA 159, it is also targeting GMAP genes in Arabidopsis. Similarly, 160, it is targeting ARF genes, 164 NAC, 165 and 166 SDG, microRNA 167 ARF again, and 169 CBF or NFYA, microRNA 171 SCL and 172 AP2 family genes, microRNA 319 TPC family, MIR 390, TAS 393 TRNF, 394 LCR, 396 GRF, and there is a lots of microRNAs still we are discovering, and we are trying to decipher the role. So this list is only for the important, showing the importance of microRNAs. Means nowadays these microRNAs are playing very important role in the plant growth and development. If we go to the particular microRNAs, if the microRNA 156 and microRNA 172 are playing very antagonistic role in the plant growth and development. So not only that, in mechanism, signal transduction, these microRNA 167 and 160 is also playing, where they are controlling the ARF genes, which are oxygen response factors. Similarly, microRNA 394, which is leaf curling response genes, they are controlling. Not only that, they are also regulating the uh, stem cells of
हाइलाइटर यूज कर लीजिए ना.
uh, 396. If we are talking about 394, 394 microRNA is regulating the Wuschel genes, which are present in the stem cells of apical meristem of plants. So these, in this way, we can find that there are lots of microRNAs, and these microRNAs are playing a major role in the plant growth and development. That's why nowadays we are very much interested in trying to decipher the role of microRNAs in plant. Now we, the main portion of the this lecture series is that the evolution of microRNAs. How and when these microRNAs evolved. If we talk about the metazoa, and after that in animals, we here we can able to see these metazoa, means sponges. They have all not having microRNAs, but they have loss of microRNAs. But if we talk about in fungi, they have no microRNAs, but they have DROSA and PASA genes. And after that divergence, when the fungi is diverged into the plant, then the DCL, HYL, and other SERET genes were evolved in the plants. So how and why these proteins which are helping in the biogenesis of microRNAs get evolved, and what are their role? It is very much interesting thing, and we're trying to find out. So there are many hypotheses regarding the origin of plant microRNAs. There are three major canonical hypotheses which are saying that plant microRNAs are evolved by the inverted duplication of target protein coding genes. Another, the first one is that they are, the hypothesis is that inverted duplication of target protein coding genes are able to give rise the microRNAs. Another one is the role of transposable elements. We are knowing that transposable elements having repeats, inverted repeats at the end, and these are very much helpful in helping to give origin of the microRNAs in the plant. And the third one is the inverted duplication of genes. Means uh, the duplication of genes, if it is duplicated in the same direction, it will not going to form a complementary part, which is which we are able to see in the stem loop structure. So inverted, if the genes are get inverted and then duplicated, they are helping into forming a stem loop structure with the complementarity. And these complementarity are not only with them, but it is also with the genes from they are arising. means the target genes. Another hypothesis is that the microRNAs are microRNAs are evolved from the duplication of pre-existing microRNAs. Means the successive mutation of these microRNAs give rise to different other type of microRNAs, and in this way they make a large family of microRNAs in the plant. And it was assumed that those microRNAs which have large members in their family, they are ancient ones. Means from if we're talking about not only the plant, gymnosperm, angiosperm, they evolved might be evolved in bryophyta, pteridophyta, and in the successive evolution of other uh, successive evolution of plants. So uh, in this way, if it is hypothesized that if the microRNA family which have a large number of members, they may be an ancient one and they will present in many plants and they are conserved. But those microRNAs which are having only present in only one organism is thought to be very recent, means they are evolved very recently and they doesn't have any homologue in other species. Another hypothesis, and what we found, the intronic regions in rice and Arabidopsis also able to help them to uh, evolve microRNAs. I'm going to give you one case study where we found that the enteric region, intronic region, as well as intergenic region have able to give the origin or evolution of microRNA in Arabidopsis. And the last hypothesis, what not it is last, but the close proximity of same chromosome forming paralogous microRNAs gene and their copies due to the tandem duplication. Means those chromosomes which are very much close to each other also having the portion of microRNAs which in the due course of evolution or with the help of duplication or tandem duplication get into evolved into the microRNA genes. So these tandem duplication sometimes arises due to the whole genome duplication, or sometimes they have get mutated, or sometimes they have some transposable elements which are moving their portion from one place to another. These all the processes help them to evolve microRNAs inside a plant. And we are knowing that plant have a very complex genome, and the duplication of genome is very high in plants, and these duplications have also helped in the evolution of many genes along with these other regulatory genes such as microRNAs and the tRNAs and similar type of regulatory small RNAs. These mutation or duplication sometimes help in the evolution of microRNA either due to the loss which is known as pseudogenes or the functional redundancy, or they are able to gain a new function which is known as new functionalization, or they have more specialized function which is comes under the sub-functionalization or functional diversification in the plants. This is the schematic presentation of how these main three hypothesis is helping in the evolution of these microRNAs. First one is the inverted duplication of the target genes. This is the one gene which is in due course of time get inverted and duplicated. So this portion is inverted and the similar. So if we get transcribed this portion, sorry, this portion and this portion, because they are inverted, must have the complementarity. After transcription, they are forming a hairpin loop where this middle portion which is not complementary to each other or the connecting portion is helping in forming a stem loop. The stem portion of the microRNA, because they are inverted, they have very high complementarity to each other. Here, the different proteins are playing major role for their biogenesis. Here, we are able to see that DCL4 and DCL3 are if processing this MIR gene, a transcribed precursor primary miRNA, is going to convert it into the siRNA, which further going to regulate the expression of target gene. But if it is processed by DCL1, the the primary microRNA is processed by the DCL1, it get converted into precursor miRNA, where the stem portion, sorry, loop portion is get cleaved with the help of DCL1, and we are getting a duplex of microRNA with some overhangs. And there is a possibility of some mismatches. If we are talking about the length of a microRNA is 21, we may get, or we found that at the position of 9th, 10th, or 11th, there may be less complementarity with the stem portion. They are forming a bulge, but this is not going to hamper the uh, control, hamper the regulation of target gene, because some because if we are talking about 20 or 21 nucleotide long microRNA, so one or two microRNA is not complementary to target gene. It is not going to disturb the cleavage or degradation of the target gene. So it is very much accepted and very much known hypothesis for the evolution of microRNAs, origin of microRNAs. Another one is the acquisition of the promoter. Means the spontaneous evolution have given small random repeats, which are going to be duplicated, and these duplication also helps them to make a stem loop structure with the complementarity. And in due course of time, because these genes which have already have promoter, their transcription also requires some regulation through the promoter region. So these genes are, for the regulation, for the transcription of these genes, there must be some regulatory portion in the promoter region. And these spontaneous evolution sometimes causes mutation or sometimes causes selection, able to give them the canonical promoter motifs which are helping in the transcription of that particular gene. So another hypothesis saying that the spontaneous evolution, where the genes may duplicated, may mutated, or may affected by the transposable elements, help them to acquire the promoter motifs which are helping in transcription. So another hypothesis is that acquiring the promoter sequences, acquisition of promoter sequence for formation of a micro, for the evolution of a microRNA. And third one, which is most accepted, is the inverted repeat transposable elements. We are knowing that transposable elements have inverted repeats at their end, and a small portion of a small portion in between them helps them to make a loop structure, because these inverted repeats are making them, and if these inverted repeats are transcribed, it will very much complementary to each other. Uh, uh, but the portion which is in between them help them to make a loop structure, which is very much similar to the microRNAs. So these are the three basic or most accepted hypothesis about the evolution of microRNAs. But not only this, we are also getting new insight after studying a particular type of microRNA. So it is not just like a principle that every microRNA is going to arises from either from inverted duplication or spontaneous evolution or with the help of transposable elements. I'm going to show you one case study. Uh, Here I'm going to show you the microRNA which is present in Oryza sativa. There are so many scientists, they have done their studies and they found that there are many microRNAs which are present in Oryza sativa, but they are not present in other animals. So here, if we are talking about rice, in rice we will found this much of mRNAs which are present, and B, these microRNAs which are present in Oryza sativa, but they evolved very earlier than rice. The rice which is comes under the monocot, and these microRNAs here in the table we can identify. B is for bryophyta, P is for pteridophyte, G is for gymnosperm, and M is for monocot. So rice is a monocot, and the microRNAs which are present in rice may evolved earlier than rice, or some of them may evolve in rice itself. So in this list, many scientists have done their research and they postulated uh, different mode of action and different time frame where these microRNAs are evolved. If we're talking about here microRNA 156, which was evolved in bryophyta, 159 in pteridophyta, again 160 in bryophyta, but 162 and 164 evolved in monocots. And and some of them are also saying that not only in monocots but they are also present in the gymnosperms. Similarly, if we are able to see this list, we will find that miRNA 397, which is present in monocot, but it is only present exclusively in Oryza sativa. Similarly, microRNA 413, microRNA 415, microRNA 416, 17, 18 to 426, and the list is going on. Means day to day, we every day we are getting new microRNAs, and these microRNAs are, if we study that particular RNA, then only we are able to know where, when, and where they evolved. So it is not a hypothesis that all the microRNAs gradually evolved, but I must say that they evolved according to their need or their requisition. Means whenever they are required in a plant, beside the other regulatory mechanism, then only the microRNAs evolve, and they help to control the regulation of target genes. Another study what I have completed at NIPGR, where I studied the microRNA 169. Again, this I found, and it is the first time we are reporting that the portion of microRNAs are also coming from the chloroplastic genome. Means the portion of chloroplastic genome, it is very well known that the gene are transferring from chloroplast or mitochondria to the host cells nucleus, and these are sometimes get integrated into the nuclear genome, or sometimes they are discarded, they have thrown out of the nuclear genome. So this is a random process where the chloroplastic genome or mitochondrial genome are transferred, lateral transfer we are saying that, into the nuclear genome. They are either sometimes they are retained or sometimes they are discarded from the nuclear genome, because due to the volume size of the nucleus, it has to maintain the size of volume of the nucleus. That's why they are not accepting more. If they are accepting more nucleotides, more DNA, for that accommodating that extra DNA, they must have to vacant some space. For that, they are sometimes throwing out some genomic part, or sometimes that chloroplastic genome out of the nucleus. But this is a random process where chloroplastic genomes are get incorporated into the nuclear genome. When we are studying about microRNA 169, what we found, it was evolved in rice only. Before, means it was first time reported in monocot, specifically in rice. And when we studied the wild varieties of rice, what we found, the chloroplastic genome, this is the whole chloroplastic genome which were available in the rice cultivar, basically in wild varieties as well as domesticated. The portion of this gene, microRNA 169 gene, have similarity across the chloroplastic genome. And the arrangement of these sequences of microRNA in the genome is similar to the microRNA. Here, the blue line is showing the microRNA 169A and its duplex part, which is complementary, which is shown in the red part. So here it is very much visible. The basically miRNA 169 is this is a precursor of miRNA 9, and this precursor have this precursor gene of microRNA 169 have microRNA 169 at this place, and its complementary part is here. So the arrangement of these two sequences in entire chloroplastic genome in such that they are aligned with the precursor microRNA 169. And when we studied about the integration of this chloroplastic genome in across the rice chromosomes, we have taken two rice varieties. One is the wild variety Oryza mariana, and another one is the Oryza sativa japonica cultivar. What we found, the chloroplastic genome are very much integrated into the chromosome number 10, 8, 1, and 4. And the portion here, red and blue portion, is showing the microRNA, which is also present in the nuclear genome of Oryza sativa. So the chloroplastic portion, which is retained by the nuclear genome, having the portions of microRNA, and they retained inside these in these chromosomes. So here we can't say that these microRNAs are arising through the inverted duplication or the acquisition of promoter or with the help of transposon, but this is a random process. But anyhow, they have a role in the evolution of microRNA. Means the chloroplast genome also have clue about the evolution of microRNAs. So this is the unpublished study, and what we found, the evolution of microRNAs not only depending upon the uh, requirement of the plant, but it is moreover required on the presence of their target genes. So here we can able to see that MIR 169 is targeting NFYA gene. So the microRNA 169 is get evolved in monocot itself, but the NFYA gene was already present in Rhodophyta very much earlier than the monocots. So it is not essential that the whenever the target gene is present, then only the microRNA is going to be evolved. It is not like that. But when the organism senses that they have extra regulatory element, then only they are uh, uh, able to means not they are on their choice, but they have some arrangement so that they can able to get microRNA, which is in further going to control the target gene. Another example is about the microRNA 167. What we found, microRNA 167, sorry, the microRNA 167 is very much conserved across the plant kingdom. Here we can able to see many different species of plants, and we found that all of them having the microRNA 167. And we are knowing that microRNAs are very much specific and very high, have very high complementarity to their target genes, and they are very much conserved. The initial hypothesis itself told us that the target genes are when get inverted, duplicated, then only we are getting the microRNAs, so that these microRNAs having the complementary part of the target DNA. So it means that they are very much specific to the target genes. But what we found in the case of microRNA 167, microRNA 167, the target of microRNA 167 in Arabidopsis and rice is ARF genes, ARF, oxygen response factors genes. But Our studies has shown that my microRNA 167 is going to regulate another gene which is not homolog of ARF, which is CNBL10. And this CNBL10, we have proved that in in Apple, these MIR 167s are controlling or regulating the uh, CNBL genes rather than ARF genes. So there is divergence, means there is a uh, there is a deviation in the hypothesis that all the microRNAs are very much conserved and they are targeting only one target gene. But here, what we found in different species, in different conditions, these microRNAs are targeting different type of genes. So one example I have shown here, it is the 167, which is a very well known fact that 167 is regulating ARF genes, but in Apple it is controlling the CNBL genes. Now the question is that Arabidopsis or rice, they are cultivated in rice is cultivated in the hot and humid conditions. Arabidopsis is grown in temperate region where temperature is very low, similar to the apple plant. The apple plants are also growing in the low temperature regions, and we don't know exact reason why the deviation in selection of target gene is in apple. But anyhow, we found that variation in the itself microRNA, variation in the sequence of microRNA itself able to change the target gene. Here, uh, this red arrow is able to show there is a deviation with the microRNA which is present in Arabidopsis, but this deviation help to acquire a different target gene in different species. Uh, Another, I'm showing you another example. Uh, Similarly, we studied microRNA, uh, microRNA 775 in Arabidopsis. The microRNA 775 was found only in this microRNA was found only in Arabidopsis, not even in the Arabidopsis lariata, which is very near to Arabidopsis thaliana. Means this microRNA 775 is exclusively present in Arabidopsis thaliana. So why this is present, why it is present in the Arabidopsis thaliana, and how it evolved as a mature microRNA. We try to find out with our canonical hypothesis. First, the microRNA 775, the target gene of microRNA 775 is the GALT9 gene. When we try to understand whether they are very much in tandem or they are very much near to each other, but it was not like that. The distance between these two genes of microRNA 775 and its target, it is a very very having 9.55 megabase pair nucleotide away from each other. Means this long space is present between these target of MIR 775. So, so how it evolved? The question is how it evolved. So when we studied about the presence of this gene in Arabidopsis genome, we did not find any promoter motif in the 5' UTR of this microRNA 775. But what we found very near to this microRNA gene, another gene is there, which is SNORNA gene. Again, but the distance between these SNORNA and MIR 775 is very less. This is 92 base pair long. But when we try to find out whether both of them comes under a single regulatory regulatory mechanism or they have separate, we did not find any promoter motive in the upstream of microRNA 775, but we found the uh, Tata box and TSS transcription start site very much in the upstream of SNO-RNA gene, and the portion between these, the adjacent gene, which is another present away from 530 base pair from this microRNA, it has 3' UTR region. This is the 3' UTR region of this particular gene, but it consists anyhow anyway they have acquired the promoter motifs, and these promoter motifs are helping in the expression of these microRNA 775 genes. So this is the another aspect where we can found that due to the mutation or due to the duplication or due to some speciation, that promoters are acquired uh, in the upstream of some portion of DNA, and these acquisition of promoter help them to express and transcribe. And here we found that this is the MIR 775 gene. So similarly, I have studied microRNA 394, which is going to target LCR gene. What we found, the evolution of microRNA 394 is in accordance with their target LCR genes. When the LCR evolved simultaneously, the microRNA 394 was evolved. So in this way, we can say that though microRNAs evolved and they have a major role in plant uh, growth and development, but their evolution is not like that. Means the theory or the hypothesis about their evolution is not like that. Ki they must have evolved. Means whenever the plant required, whenever the plant having, they want to get some extra regulatory element, they have acquired anyhow the evolution of microRNA, microRNA, RNAs, and other type of RNAs, RNA, which are going to help them their expression. So in this way, I'm concluding my topic, and I would like to acknowledge and thank my director at NIPGR, Dr. Anand Sarkar sir, and the team members uh, which were present there, and they helped me a lot to understand the microRNA, and I supported their studies by finding the evolutionary analysis, and separately, individually, we can able to understand the evolution of microRNAs, which are very much a important part of a plant growth and development.
Thank you